Hi, welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. In this examined content, we're going to be dissecting this particular paper, looking at how tumors, cancerous tumors, can self-seed themselves as well as seed other tumors to grow at a far faster rate than they normally would, which is an absolutely incredible phenomenon that occurs with cancer. So if you'd like to learn about this particular process, then in this brilliant paper, that's what we will be discovering. So without further ado, let's jump into this examined content. Okay everyone, let's jump into the experimental method, the design of this particular study. It's going to be an involved one. I'm going to be saving particular chunks of the experimental methods for the actual figures because they are so involved. But overall, we're going to be looking at a lot of mouse models of particular cancer types. So these are mice and cell types that have been manipulated to express particular cancers so that we can study those cancers in a living tissue. Meaning we have cancers that are very low metastasis level, or they're very high levels of metastasis, as well as in different tissues. So you're gonna have low for skin, high for skin, you're gonna have low for breast cancer, you're gonna have high for breast cancer, and a bunch of others as well. So there's a lot going on here, and again, of course, I'll detail this stuff in more detail when we actually get to those figures so that I'm not bombarding you with a bunch of information right now. They used in vivo systems, meaning that they used mouse models to show these different cancers. They used ex vivo systems where they removed the tumors out of those particular mice and then did experiments on them. And then they also did in vitro models where they looked at particular cells. So they did a large number of different studies. They did a lot of fluorescent microscopy, meaning that they used microscopes to be able to investigate inside the tissues based on fluorescence of particular proteins. They looked at gene expression, they looked at mRNA, and I'm going to leave it there because again, I think it's for this kind of paper, it's absolutely more beneficial to get into the figures where I can actually show you specifically what they're doing for each particular experiment. So without further ado, let's jump into the ever extremely exciting results of this paper. Okay, so for figure 1A, they're showing just a diagram of what they did for the actual experiment. So for figure 1A, they're showing that they have these mice and they're injecting a recipient tumor. So they're injecting a particular amount of cancer cells, which is a relatively large amount of cancer cells, probably in the millions or 500,000 or something along those lines. And those are what are considered unseeded tumor cells. So meaning that they are not tagged in any way. We just know that they're going to grow into a tumor. Now on the other side of the mouse, so they just move to the other side of the mouse, they inject a donor tumor which is going to express a particular fluorescence. So then they can track the cells from the donor tumor, and if those cells migrate to the other tumor that doesn't have any sort of fluorescence on it, and they're able to track that through, of course, the fluorescence of the cells in that donor tumor. So that's exactly what they're doing, and that's exactly what they're showing with figure 1A. Now let's look at the actual data. Now 1B is where they've excised the tumors. They've taken the tumors out of the mice after a set amount of time, something like 8 weeks or some particular amount of weeks, and then they're looking at the fluorescence. Again, how much of the donor tumor has then migrated or metastasized or moved, or the term they use is seed, has seeded into the other tumor, the recipient tumor. So it's called a recipient tumor because it is is presumably going to accept cells from that donor tumor. And as you see more fluorescence, that means that that donor tumor on one side of the mouse has given its cells to the other tumor, which is crazy to think about. Already we're getting some really interesting data here. So let me run down what all these different cancer types are. The LM2 is a lung cancer marker. So here you're seeing great expression of this seeding from one tumor to the other tumor. The MDA231 are also lung cancer cells, but these are unmarked. And you're seeing essentially, so what you want to do is compare that to the LM2, which are both marked, and you'll find that there's essentially no difference. So there's a mass migration of these marked cells from the donor tumor to the recipient tumor. Now for the next two, they are changing what type of cancer cells that they're injecting for the donor tumor. So for the first one, the CN34BRM2 are brain metastatic cancer, and the other one, the MCF7BOM2, are bone metastatic cancer cells. And of course, you're seeing lower levels of that seeding from the donor tumor to the recipient tumor, but it's still occurring. And then the final one is where they've changed the recipient tumor just to see if a different type of tumor would have an impact in terms of this migration from the donor tumor to the recipient tumor. So the recipient tumor has been changed to a low metastatic cancer cell. MCF7 is a low metastatic cancer. So, and yet we still see some of that seeding that does occur. As a matter of fact, although the tumor itself, the size isn't that great, the vast majority of that tumor comes from the donor tumor. So that means that the cancer cells, the highly metastatic cancer cells that are coming from the donor, are populating this lower metastatic cancer to such a great degree that it's essentially overcoming the fact that it's a low metastatic cancer, as it's starting to really build up the density of those donor cells. And then for figures 1D and E, for D, they're looking at the donor tumor, so the one that has the injection of these highly metastatic cancer cells, and the green fluorescence is those particular cancer cells. Now, the interesting part is looking at the recipient tumor, which is now fluoresced in red, so that would be the presumably the less metastatic cancer, or just the tumor itself. And any green that you see there has come from the other tumor, meaning that you have this migration because it has to move through the bloodstream, it has to move into this new tumor. So here you're seeing this invasion of this heterogeneity of these different tumor cells that are starting to mix and match because of this donor tumor. And then looking at figure 1F, they're redesigning their experiment. So in this situation, what they're doing is that they're injecting cancer cells into the tail of these mice, and then that is actually leading to a lung cancer phenotype. So it's showing cancer or starting tumor progression in the lungs, and then after a set amount of time, once that tumor has taken hold, then they inject another tumor into another site of the mouse, just to see if these circulating tumor cells will actually still end up invading this new tumor that they're forming in the mouse later on. And what we find is that with G, there's more and more of that invasion of this new tumor is perfectly proportional to the amount of circulating tumor cells that are found circulating in the bloodstream. So there's a proportional association between the circulating levels of these tumor cells with the amount that's found in the recipient tumor, even though that recipient tumor wasn't injected till weeks later. Now for figure 2, they're doing the exact same thing they did for the first experiment in figure 1, where they inject a fluorescent tumor the donor tumor into one side of the mouse and then a recipient tumor that does not have any sort of fluorescent cells, just to see if there's that cross-contamination from those circulating tumor cells that would come from the donor into the recipient. So they allow all that process to happen, and then what they do is they excise the tumors, so they take the tumors out, and then they separate out the fluorescent cells, so they want to get a specific look at those cells that move from the donor tumor, because there are still cells in that donor tumor. So what about those cells, which are now called S1 cells, those S1 cells led them to leave the donor tumor and go to the recipient tumor? What is so special about those cells that's leading to a cancerous invasion of the other tumor? And they show for 2E a gene profile of a comparison between different types of cancers. So they've got these S1 cells, which are S1A and S1B, as well as parental MDA231 cells, which are also a cancer type, but they are low metastasis. So the S1 are presumably high levels of metastasis, and the parental cells are low levels of metastasis. And that's actually going to continue on for the rest of this paper. So keep that in mind. If you see parental, that means low metastasis level. And then not only that, they compare that to other forms of metastatic cancers, and they show the changes in gene profile. And you do see that there is a difference in the different genetic markers that they did look at. So different genes are expressed for high metastatic, moderate metastatic, as well as low metastatic cancers. And then for G and H, they're taking these S1 cells, which again they've isolated from the tumors from before, and they're re-injecting them into mice, and then they're seeing how metastatic these cells are compared to low metastatic cancer cells, so the parental cells. And what we find is that naturally these S1 cells lead to mass metastasis compared to the parental cells. The parental cells are taking much longer to become metastatic and spread over the mouse. It's to the point where you can't even detect the levels. And they're looking at bone as well as in the brain. And in both, the S1 cells are extremely metastatic. All right, now I'm skipping figure 3 because they are repeating a lot of experiments that we saw in figure 2, but they're injecting it into the heart specifically as opposed to the tail, which is what they used earlier. But they show the exact same results with that figure as they do with figure 2, so I'm not going to cover that again. However, figure 4 is where they're doing a migration assay or a transendothelial migration assay, and this is essentially an experiment where you put the cancer cells and separate them with an endothelial layer, which is supposed to be a representative layer of your vasculature, of your bloodstream. The lining of your vessels is made up of endothelial cells. So if you have this migration through the endothelial cells, then that means that it's starting to get into your actual tissue. And that's kind of the initial stages of metastasis. So they plate the cancer cells on top of this endothelial layer, and then underneath the endothelial layer, they have different media. So they have different, what's supposed to be representative of blood. So the different factors that might be in blood, so they're going to switch out those factors. And then they're going to be able to see which one of those factors, or a series of different factors, are going to impact the cancer cells to drive them to go into a metastatic state where they move through that endothelial layer. So what do we see with 4B is the use of 2 different cancer cells. So one is high metastatic and the other one is not so high metastatic. So one of them is a skin cancer, a melanoma, and the other one is the typical MDA cells that we've been looking at, which are highly metastatic, especially the LM2 variety. And then the only thing that they're actually changing is the media. So they've got 3 different media conditions that they try, and then they want to know how metastatic do these cells react. So if they use MCF10A media, that is serum levels that are absolutely normal. So that's what you would find in a normal individual that doesn't have any sort of cancerous markers of any sort. And you see that the migration, otherwise known as the metastasis, that beginning section of metastasis, is impaired. So it's certainly much slower with that media. However, if you look at A375, which is media that's taken from a surrounding of low metastatic cancer, you see that there is an elevation in that metastasis. And that's with both of the different cancer types. So if that's the melanoma, which that media corresponds to, or if you're looking at the LM2 cells, those also increase their metastasis in that situation. So the media clearly makes a difference, but it certainly makes a difference when you link up that particular media, the A375 media, with the A375 cells. They become more metastatic, maximally metastatic. And then you see the exact same result when you look at the LM2. So when you have the MDA231 media with the MDA231 LM2 cells, because those correspond together, you see the maximal levels of metastasis. And of course, by comparison of the A375 melanoma, the low metastatic cells, those show decreased levels of metastasis, but certainly still elevated compared to the normal media condition. So if you have high metastatic cancer cells in a high metastatic media or blood serum, you're going to have the highest level of metastasis. And if you have high metastatic cancer cells in a low metastatic cancer environment, then you're going to see lower levels of metastasis, but you're still going to see metastasis. And in all conditions, you are going to see some level of metastasis. It's just the level of that metastasis is modulated by the media that the cancer cells are in. Now for figure 5, they're trying to figure out which kinds of cytokines, so communication proteins between cells, modulate this metastatic communication between these cells. So they're using a cytokine antibody assay. So there's antibodies to every single cytokine, but they're separated out into a bunch of different wells. So then if you add the serum of those cancer cells to that particular assay, to that particular plate, then they're going to be able to tell how much of each antibody is bound to a particular cytokine. So of course, if there are no cytokines found in that serum, you're not going to see any expression. You're not going to see any black dots. But if there's a lot of that cytokine, then there's going to be a lot of binding and you're going to see a lot of expression. That makes a lot of sense. So then they compare 3 different lines of cells. The first one is normal cells. So these are non-cancerous cells. And the other 2 are cancer cells. So the MDA line, which is what they've primarily looked at so far. So the MDA line, again, for the normal ones are the lower metastatic level, but the LM2 are the higher metastatic level. And there are similarities between the 2 when it comes to the cytokine assay, which are not found in the normal epithelial cells, the breast epithelial cells that are normally functioning and are non-cancerous. So by comparison of all 3 plates, you can tell that there's 2 dots there that are expressed in the cancerous cells that are not expressed in the normal cells. They investigate those 2 communication proteins, and those 2 are interleukin 6 and interleukin 8. So then they isolate those 2 and then they add those to the endothelial assay that I mentioned earlier, where they see this transmigration, this invasion through the endothelial layer as a kind of a marker of this metastatic potential. And what they find is that there's a greater invasion with the addition of these interleukins. So if it's interleukin 6 or interleukin 8, you're going to see more metastatic potential in the cells because of the addition of just these cytokines. And then they go back to one of their older models, which they showed in some of the previous experiments, and they want to know if they knock down the effect of IL-6, so they essentially take the receptor that's found on the cells and they inhibit it so that it can't bind that IL-6 cytokine on these cancer cells, do we see decreases in this seeding from the donor tumor to the recipient tumor? And what do we find? Yes, there are decreases in this seeding behavior. So clearly one of the mechanisms for this seeding, this circulating tumor, where you have this migration from donor tumor to recipient tumor, is at least partially mediated by IL-6. And now in figure 6, they're trying to find out what happens to the size of the tumor, the actual volume of the tumor, when you inject these highly metastatic cells back in. So here they're injecting the tumor, again waiting a particular amount of days, and then they're injecting the fluorescent tagged cells, the cancers, the highly metastatic cells, and they want to know how that impacts the volume of the tumor over a set amount of time. And then what they show is that there's a massive increase in the size of the tumor just by the injection of these highly metastatic cells. So the low metastatic cells show a slow rise in this tumor, but when you inject the high metastatic cells, suddenly it increases way faster than if you just had the low metastatic cells in that area. And then D, they're doing fluorescent microscopy. So they're essentially looking and fluorescing the blood vessels that would be found in that tumor, and they find that between the unseeded, so that would be just the regular tumor without the addition of these highly aggressive cancer cells, and comparing that to the seeded, where they do add these CTCs, these circulating tumor cells, and you find that the blood vessels, the amount of blood vessels, increases substantially. So the tumor has to feed itself. So what it does is it allows for this proliferation of more vessels around it that will integrate into the actual tumor itself to feed it a greater blood supply, more oxygen, more nutrients, things of that nature, potentially more interleukin-6, interleukin-8, things of that nature, different factors that will allow it to grow and grow and grow. And then E, F, and G, they're showing different microscopy images. The green is again the tumors that are seeded, so those CTCs, the circulating tumor cells that are invading into that particular area of the tumor, and the red are signals of or protein markers of immune cells. So you're seeing some of those immune cells infiltrate into the tumor, trying to deal with the issue, trying to eliminate all these different cancer cells that constantly are proliferating. And finally, some additional notes that they mentioned in the body of the paper is that, well, one is that these CTCs, these circulating tumor cells, do not create their own tumors. So they only are released and only act on existing tumors. So there has to be a tumor where they come from, and then there has to be another tumor that has formed somewhere else, so the malignancy has already started to happen, and then they infiltrate this new tumor and then make it grow and proliferate much, much quicker. But do they themselves create their own tumor? No, not according to these researchers. Another really interesting point is that there is cross-contamination. So just because they come from, let's say, a breast cancer line, that does not mean that they will not infiltrate and make another type of cancer or tumor grow. So it could be breast to like a skin cancer. That would be no problem. The CTCs would still function in that regard. And then the final point is that these CTCs secrete an attractant, a chemoattractant, a cytokine for themselves that's called CXCL, which is something that we also create, our cells also create, but it attracts immune cells. So the tumors will attract immune cells to themselves because the immune cells produce interleukin 6 and interleukin 8. So they release, these immune cells will release IL-6 and IL-8 because we also use those particular cytokines to recruit more immune cells. However, the danger of that is it's a cyclical pattern. The CTCs will recruit the immune cells, and then the immune cells will, not because they want to, but because they're trying to recruit more immune cells, they end up recruiting more CTCs. So it's a vicious cycle, which is absolutely mind-blowing, how the tumor is trying to attract the thing that is trying to kill it because it can then grow at a faster rate. So it's a small amount of damage it'll take just to eventually grow stronger. So what are the big takeaways from this massive paper, incredibly interesting paper, is that there is such a thing as circulating tumor cells, and tumors will share their cells, will cross-contaminate one another, which leads to a massive spike in their speed of progression, which is crazy. Not only does that happen, the CTCs themselves are not metastatic in terms of they don't create their own tumors, but once a fellow tumor, a cooperative tumor has occurred somewhere else in the body, those CTCs will migrate to that tumor and massively expand that tumor to a point where it becomes incredibly dangerous. And then they secrete different chemoattractants, will attract our immune cells, which then will attract more CTCs through their own chemoattractants. And some of those chemoattractants were, as we discussed, interleukin 6 and interleukin 8, but surely there are others other than just those two. Not only that, CTCs and tumors in general will metastasize regardless of their environment, so it doesn't really matter about the serum, but the environment that they're in will impact how quickly they metastasize. They won't dictate if they metastasize, that's dictated by the cancer cell itself, the genes and the overall expression, the type of cancer it is, but the environment will impact how quickly that cell is able to metastasize and move to a different location and then grow into a tumor. And in terms of this cross-seeding, this cross-communication between tumors, it is not dependent on the type of tumor. So you can have a breast cancer that will cross-contaminate and cross-seed with a skin tumor or a brain tumor or a bone tumor. It doesn't matter. As long as they have access to one another, which they will have, then they will cross-contaminate, leading to a much faster speed of growth than you would if they didn't do that seeding procedure. So, I mean, this is crazy. This is really, really cool research. Obviously, it's more informative in terms of like how we stop these CTCs, how we stop these circulating tumor cells is still to be found out, but it is incredibly powerful to know that this cross-communication between tumors and how they modulate our immune system leads to this incredibly fast progression of cancer and metastasis. Okay, well, hopefully you found this informative. I really hope that you did. It was a monster of a paper, as most of these cell papers tend to be. And with that said, I hope I have the absolute pleasure of speaking with you in the next one. Have a good one, guys. See ya.